The story of charge transport in DNA started about 20 years in the context of molecular electronics. And at that time, the big hope was that it will lead to computers or electronic circuits that may compete with the microelectronics industry in view of the fears of the breaking of Moore's law. And over the years, this did not happen, but still the question of charge transport in DNA raised again and again, and the contradictory results were received. And the main reason is because these experiments are difficult. And it is difficult to measure not millions or hundreds or thousands of molecules, but just to measure one molecule again and again in a reproducible manner. And that's, in fact, the big breakthrough that we have received over the last years. And about a year ago, we were lucky enough to have a publication in Nature Nanotechnology where we reported a reliable charge transport in DNA, and we could go forward and explain the mechanism. So the main actor in this work was a former PhD student in my group, Roman Joravel. He's doing now a postdoc in the US. So why DNA electronics? You're all familiar with this molecule. And this was in this context, not in the biological context, but in a context of molecular electronics that deals with the electronic properties and the functionality of single molecules. And indeed, DNA, because of its double-strand recognition and the ability to form complex structure, was a natural candidate. One thing is that it's very controllable. You can determine the exact molecular sequence and the structure. The self-recognition and the self-assembly allow complex structures predetermined, and you can form very complex structures like those shown here. However, contradictory results were obtained in charge transport measurements, but the exact transport mechanism was still unclear until these results. And now when we look carefully at the structure of DNA, it is made of 4 bases: adenine, thymine, guanine, and cytosine. And that's usually the components that we look at, and we kind of ignore the backbone. This is a sugar phosphate backbone that is actually forming the external part of the double helix, and is very important for our talk, and not only the core of the bases. And here comes the question: if DNA is to conduct, where would the current flow? Would it be in the core of the molecule through the bases, or would it be in the backbone, in the periodic structure, which is surprising because these are sigma bonds. And the most common assumption was that charge transport goes through the bases. And the next question that is coming is, what is the exact charge transport mechanism? And one of the important context to explain charge transport is the context of localization. When we look at a single molecule, we can see that the orbitals over the molecule may spread all over the length of the molecule. And in this case, it will be fully delocalized, or they can be isolated in specific places along the molecule. And in that case, charge will have to hop from one orbital to the next and to the next, and so on. So when you think about the possible mechanism in this regard, then the first would be coherent mechanism, which is usually called tunneling or super exchange. And this will be dominant in short molecules. This will not be temperature dependence. Usually it will support higher currents, and it will require the localized orbitals. So in this case, there will be kind of a one jump tunneling through the system from one side to the other, with a functional form in which the current decays exponentially with the distance. The other common mechanism is incoherent. It's called hopping. And in this case, the charge is hopping from one side to the next and so on. This will be dominant in long molecules. It will have strong temperature dependence. And in this case, usually there will be a lower current. And this will be in the case of the localized with a functional form that depends on the activation energy and reversely on the temperature. And that gives us, these two functional forms give us a handle to identify what is the actual mechanism of charge transport. And this may, of course, combine a contribution from both types and from other physical phenomena. Now, when we want to identify the mechanism, the question is, what do we need in order to identify this mechanism? So the first information would be current voltage, IV, measurements that will tell us if the molecule is conductive and in which conditions. The temperature dependence can tell us if it is coherent or incoherent, if it is thermally activated, and what would be the activation energy. And if we use different sequences, it will tell us if the base sequence or the type of bases is important for this mechanism. And finally, gate measurements that I will immediately explain will tell us if the contacts are good, if we have charging effects, what's the density of states, and so on. So just to explain the concept of the gate, we usually think about two electrodes, source and a drain, and an object, the molecule, a nanoparticle, in between that we want to measure. So when we apply voltage, current will flow from one electrode to the other, to the object, and then to the other electrodes. So when we perform current voltage measurements, so we apply voltage or we change the chemical potential or the Fermi energy in between difference between the electrodes or in one of the electrodes, then we raise the voltage until we hit the position of certain energy levels. In the current voltage scheme, there will be no current until we hit this energy level, and then there is a rise of the current versus the voltage. Then we raise the voltage again until we hit the next energy level, and then we have again another rise of the current, and so on and so forth. And if we look at the derivative of the current versus the voltage, we will have these peaks that will tell us the positions of those energy levels. This is the measurement when we have just two electrodes. But we can have a third electrode, which we call a gate, and this electrode is not electrically connected to any of the other two electrodes, the source and the drain. So there's no current flowing between the gate and these electrodes, but there is an electrical potential which is applied and affects the object between the electrodes. So in this case, when there is some voltage difference, usually small, then the application of the potential here shifts the energy levels and shifts those position of these two curves, and it can provide us the information on the density of states, and if there are charging effects, it will tell us how good the contact is between the object and the two other electrodes. So, and here I'm going to overview the previous experiments that were done before. So the first one, and here there are thousands of experiments that were reported, the original papers in these fields, these are cited between 1,500 and 2,000 times. So I took really the three pioneering ones. And the first one was an experiment that was done in the Technion in Haifa in Israel. And in this case, they took a 10 micron long DNA molecule connected to two metal electrodes. And what they measure is shown here. This is not just the x-axis. This is no current whatsoever. In fact, this inset is probably responsible for the majority of the citations of this work. So no current was flowing when a long molecule, DNA molecule, was on the surface, coupled to the surface, and voltage was applied. Soon after, there was another report by a group in Basel, by Fink and Schonenberger, and they showed that when you take 100 nanometer long, hundreds of nanometer long DNA molecule between two electrodes, a tip and a metal grid, apply the voltage, then it seems like an ohmic behavior. This was not, a result was not repeated as is. There were probably some problems with the experiment, but these were really two extremes. And this was the background for my decision to join this field. And this was resulted in my postdoc work with Case Decker at Delft. And in this experiment, we decided to measure a short 10 nanometer with 30 base pairs long DNA molecules, all homogeneous, poly G, poly C, and we trapped them between two electrodes in the scheme that is shown here. And we could show with the help of a lot of controls that a current could flow between through the DNA between the electrodes. But the drawback in this experiment was that we could not see the molecule. It was very hard to verify that there is indeed just one molecule, and we had to use a lot of additional controls in order to do that. And we really wanted to make a better experiment, an experiment in which we measure just one molecule. We can see that the molecule is there and verify by alternative manners that the molecule is there. And finally, we wanted to measure it again and again and change the conditions and see how the transport behaves. So the charge transport in any case was strongly dependent on the molecule and the experimental system. So that was the background to the experiment that we want to do now. But what do we know from the previous experiment? And this was summarized in a review in advanced materials that we prepared, cited here in the bottom. And we found first that significant current is transported in short DNA molecules, and that depends on the experimental system. No charge transport was going in long DNA molecules that were deposited on a hard substrate, so the coupling to the solid surface decreased the conductivity, and also that measurements of 10 to 100 nanometer DNA are very challenging. Most of the experiments published before were done either on shorter than 10 nanometers, usually in break junction systems or STM, and longer ones were done on hard surfaces. The most common charge transport mechanism was thermally activated hopping, and most of the mechanisms did not involve the backbone. So the question was, what is needed in order to do a good experiment in which we know we address all the challenges that I mentioned before? So the first requirement would be to have exactly just one molecule in the junction. In addition, we want to have complementary characterizations. So another way by AFM or by SEM or by another way where we can see that the molecule is indeed there. In addition, we want an isolated molecule. So we want to avoid interaction with the surface that, as I explained, may decrease the current. And we want to have good contacts to the leads. And we want to have a stable junction. So unlike the STM break junctions that you measure a certain molecule, but you cannot really address a specific molecule, we want a molecule that can survive long enough, and we can perform repeated measurements on this molecule. And if we fulfill all these conditions, then we can perform IV measurements, gate and change the temperature, measure the temperature dependence, and we can change other parameters like humidity or the measured sequence, and so on. So that's kind of the requirement for what we term as the reliable and good experiment. So how did we do that? You have the molecule, and then we want to trap it between electrodes, apply voltage, and measure current. Easier said than done at the nanometer. So the experimental system that we chose is two metal electrodes that are fabricated by e-beam lithography, where in between we have an object that we call a dimer. This is a single double-stranded DNA molecule connected to two nanoparticles by tiles or by other means. And the dimer is connected to the tail electrodes and raised above the surface such that we can perform good measurements. These nanoparticles can report and tell us that the dimer is indeed trapped between the electrodes. So that's very important. And also, they provide good contact to the electrodes. And here you can see the experimental system. Here you can see the chip with a lot of electrodes shown here. And somewhere in between the electrodes, we have these two nano electrodes with 30 nanometers in between. They can be seen here in the AFM image, and there we can perform the measurements. So how do we make sure that we have just one molecule? So we mix nanoparticles, in this case, 8 nanometer particles with 100 bases long single-stranded DNA with a tile in the end. When we mix them, there will be some connections. Some nanoparticles will remain without DNA. In some cases, we'll have just a single DNA strand that binds to the nanoparticle. And in other cases, there will be 2 or 3, and so on. So that's determined by the relative concentration. Then we can run the solution in gel electrophoresis, and then we can see that the first band will be the single particles. The next band will be nanoparticles with a single DNA strand, and the next bands will be 2 and 3 DNA strands that are connected to the nanoparticle. Then we can extract, we can really cut the band and extract the structures where you have a nanoparticle and a single strand. We can do the same with the complementary strand, and in this way we get the dimers with a single DNA molecule connecting the two nanoparticle. And indeed, when we deposit them on the surface, we can do a scanning electron microscope imaging, and we can see that most of the structure are indeed dimers. If we take the first band, it will be just monomers, and if we take the upper bands, there will be more complex structures. So how do we position the dimer between the electrodes? It's not as easy as at the micro scale. We apply an electric field between the two electrodes. The field is not uniform, and it applies a force on the dipole that is formed in the dimer. And then this is the formula that determines the trapping. I will not go into all the details, but the left term includes the radius of the nanoparticles, so that's the size of the nanoparticle. The right-hand term refers to the field magnitude and the gradient that is applied, and the middle term tells us the dependence on the frequency and the composition of the solution. So in this way, we can control all the parameters that are needed for the trapping. So when we test the effect of the size of the nanoparticle, we took a solution in which 99% are 8 nanometer nanoparticle. GNP stands for gold nanoparticle, and 1% or 20 nanometer particle. And after applying the voltage, we can see that most of the trapped particles are the 20 nanometer particle. So in this sense, the larger the particle, the easier it is to trap in between the electrodes. But we want to have a matching between the size of the particle and the DNA, so we chose to work with all 8 nanometer particles. Then we test the effect of the solution composition. I will not go into all the details, but here we compare two solutions, and we can see that in one of them, there are too many nanoparticles, and in the other, there are no particles at all. So that's very important to control the composition of the solution. We then went forward and tested the shape of the angle, the shape of the electrodes, and we could see that there was no big effect on the trapping, and what is actually determining the trapping is really the tips of the electrode, which requires more sophisticated nanofabrication or e-beam fabrication. And finally, we saw that at least in the range of 0.1 to 80 megahertz, there was no big dependence on the frequency. Then we tested the effect of a gate. In this case, we wanted to check the system. So we trapped a single 20 nanometer gold nanoparticle between the two electrodes, and we could see that the stability diagram, the voltage, the source drain voltage versus the gate voltage. And here you can see the derivative of the current, and these diamond-like structure tell us that indeed the Coulomb blockade is appearing and the junction works well. Now, and another information that we got is that the gate is sufficiently effective and for the measurements. The experimental system is shown here. This is the scheme that I showed before. And here you can see a scanning electron microscope imaging of the electrodes. That's before the trapping. So the electrodes are empty. And after trapping in the way I described before, you can see that a nice dimer is trapped and nicely aligned between the electrodes. And then we could do the experiments. So we have, as we asked before, just one molecule. We have good contacts. The molecule is suspended, so there's no contact with the surface. We can see that the dimer is there, so that's the additional way to verify that the molecule is there, a single molecule, as we showed by the preparation and separation by the gel. And indeed, we have high experimental confidence when the price is that the statistics of current voltage is much smaller than, for example, in STM break junctions, when you can measure thousands of curves again and again and again on many different molecules. So the first molecule that we measured was what is called random sequence. It's not random. We chose it such that it will not fold on itself, but it contains all the four bases. And this is probably the most important graph that I show in this part of the talk, where you can see that we can get current of up to 10 nanoamps. We limited the system to 10 nanoamps to avoid burning the molecule. And we can get kind of a smooth and linear or S-shaped curve. And that shows that a 30 nanometer long, 100 base pairs long DNA molecule can conduct significant current. And that's very important. It addresses the question whether DNA can conduct. Yes, it can conduct significant current. This was measured at 5 Kelvin. Then we went forward and we looked at the gate effect, and we can see that it is quite smooth, different than the diamond structure that we saw before. And that indicates that the contact between the dimer nanoparticles and the electrode is very good, as we wanted to have, and we don't have a Coulomb blockade effects. So this is the scheme that I mentioned before. The good contact is here and between the molecule and the nanoparticles. Then we went forward and we measured a homogeneous sequence. We naively assumed that we will get a better current because it's all guanine bases with the lowest oxidation potential. And also it is homogeneous, so the levels could be aligned. And surprisingly, we got currents that were roughly similar to the previous measurement. So that was very strange and unexpected. So then we checked the temperature dependence to address the mechanism of hopping versus tunneling, as we mentioned before. So you can see that we start, this is the colors are representing the temperature starting from 5K and up to room temperature. And you can see that the lower temperature up to 50 degrees, there isn't much change in the current. And then after 50 degrees, there is a start of a decreasing of the current to lower currents, usually at room temperature. And similar behavior was also observed for the homogeneous poly-G, poly-C DNA. And if we look at the cut, a vertical cut of the current versus the temperature, we can see, and here in this Arrhenius plot, we present the log of the current versus 1 over the temperature. And we can see indeed that the current is not changing much up to 50 degrees, and then there is a slow reduction of the current. So to conclude this, the experimental observations, there was no temperature dependence up to 50K, and there were high currents at the low temperatures, and that means there's no thermal activation. It cannot be hopping and thermally activated hopping. And then the other observation is that the random sequence and the poly-G, poly-C behaved similarly, means that it's probably not a conduction band due to pi-pi stacking, and that was hitting to the possibility that the backbone is transferring the current. That was very surprising for us, especially that the backbone is made of sigma bonds, but we went on to test it experimentally. So again, when we look at the structure of the DNA, we can see the backbone and the bases. And in order to check if the backbone is conductive, we can simply cut it. Cut it means that one phosphate is missing. This is called a nick in the backbone. And that will enable us to see, because if we cut or make a discontinuity in the backbone, current shouldn't transport in the backbone. And when we measure if the current is going through the bases, we would expect current to flow even in the presence of a nick. And in the absence, if the current is going through the backbone, then the cut will reduce the current. So we went, and these are the results that I showed before. In a regular dimer, either homogeneous or non-homogeneous, we got a similar current. And when we check the DNA, and here we made a nick in each one of the two strands because of the double helix, we wanted to cut both strands, and we did it, the nicks far apart. So there is about 50 nanometers between the two nicks, so they cannot affect or form some kind of disruption to the structure. And then in this case, there was no current at all. So that was a strong hint. Now we wanted to check if the nicks disturb the transport in the core of the DNA. So for that, we made only one nick, and the current was similar to what we measured before. So that means that the backbone plays a crucial role in the transport, and even a single discontinuity in each one of the strands collapses, suppresses the current. So when we look at all the data, and here you can see all the data except for the 0 in order to avoid a lot of yellow here, we can see that there is some variability, which is not surprising when you measure single molecules. These are the results up to 50 degrees. It seems like maybe there are some preferred patterns here. Now, here you can see the data also for the poly-G, poly-C, and for the one nick, and you can see that the general behavior is generally the same. And now we wanted to understand what is going on, what is the mechanism, and we collaborated with Professors Spiros Cortis from Cyprus and Carlos Cuevas from Madrid, and they made, together with some other collaborators, they made a calculation, and you can see the presentation of the HOMO orbitals on the molecules. In a 1D presentation, you can see the density of states versus the energy. The green here is the Fermi level, and you can see the base pair density of states and the backbone. So you can see that the bases are closer to the Fermi energy, but also then appear the backbone states, and they're smooth. So there are states for both the backbone and the bases. Now, when checking the localization length versus the energy, the localization length was very, very short. And that means it cannot be, as we thought when we looked at the experimental results, that there is some kind of band conduction. It must be some kind of hopping, but not the thermally activated hopping that we were looking for before. So when we try to understand the meaning of the results, we can see that the backbone is periodic. So we have nearly similar states because of the structure, the physical distortion, and the temperature. There might be some small difference between their positions. So when we raise the voltage and we open some kind of a window here, the temperature causes some kind of resonant hopping through the backbone states from one side to the other. And at higher temperatures, there are vibrations, and they cause the reduction of the current, because the resonance is not as good as before. When we compare to the base states, so in the case of the random sequence, the bases are not aligned and they kind of flicker. So then there is much less possibility for such a resonance, and then we don't have the current through the bases. So that's kind of explaining the experimental behavior that we see. So to summarize, a single molecule experiment on 100 bases, that means 30 nanometer long double-stranded DNA, confirmed that there is a single molecule, and we could measure again and again the same molecule that had good contacts to the electrodes. We had high current at low temperatures and constant current that decreased with the temperature above 50 degrees. We did not observe a sequence dependence, and that suggested after the measurements with the nicked DNA that resonant hopping through the backbone might be the transport mechanism. And then we looked for what can we do with these measurements, for example, for sensing. So you can see here that there is a dimer trapped between the electrodes with a missing part in the center. And when you bring a DNA which is complementary, then the current will turn on, and that may be relevant for DNA, for RNA. So it's relevant in the context of corona or in the context of cancer. So you can see here a preliminary experiment that supports this assumption. That's the DNA with two 40 bases on the sides and a 20 bases segment in the middle, which has two nicks on both sides. And when then we raise the temperature and get rid of the middle segment, the current is suppressed. And when we expose it to the segment again, we get a recovery of the current. And here you can see the dimer trap between the electrodes. There is another dimer, but it's connected only to one electrode. And so this was done in collaboration with Sasha Kotlia, who prepared the poly-G, poly-C dimers with Spiros Cortis and Carlos Cuevas that supported the theoretical calculation. And I would like to thank again my students, and especially Roman Juravel, who did most of the activity in this experiment. And I would like to thank you for listening. Thanks.